Okay, let's unpack this. Imagine tiny, potent signaling molecules born right from the membranes of our cells that can orchestrate everything from the silent ebb and flow of blood pressure to the intense coordination of childbirth or even the precise inflammatory response when you get a cut. Sounds like something straight out of a, biological control center, right? But it's happening inside you right now through an incredible dynamic family of compounds we call eicosanoids and platelet-activating factor, or PAF.
Precisely. These aren't like hormones that are pre-packaged and stored. They're created entirely on demand, which makes them incredibly precise tools for the body's immediate localized needs. Think of them as a highly specialized rapid response communication system. Today, we're taking a deep dive into the fascinating, intricate world of these lipid-derived autacoids, drawing from the authoritative insights of GOODMAN and GILMAN's The Pharmacology. Our mission is to really master this information, gaining an expert-level understanding that will truly deepen your grasp of human physiology and pharmacology.
That's our challenge today. We're going to pull out the most important nuggets from this dense, rich material, but we're going to go beyond just the surface. We'll explore where these molecules come from, how their complex biosynthesis is orchestrated, how they communicate within your cells, their surprising and diverse effects across nearly every system in your body, and crucially, how we've learned to harness or block them therapeutically to treat disease. Get ready for some truly illuminating aha moments that will connect the dots in ways you might not have expected. So let's start at the beginning. What exactly are eicosanoids and PAF, and what makes them such special messengers in our bodies?
Well, at their very core, eicosanoids and PAF are crucial signaling molecules. We often refer to them as autacoids, which comes from the Greek for self-remedy, kind of indicating their localized action. They are derived directly from lipids within cell membranes, primarily from a 20-carbon fatty acid you might have heard of, arachidonic acid, or AA. What truly sets them apart from traditional hormones is that they are not stored in advance. Instead, they are synthesized rapidly and on demand. Most cells in the body can produce eicosanoids, while a more specific, smaller set of cells produces PAF. This rapid localized production is triggered by a wide array of stimuli, anything from a physical injury to chemical signals or even hormonal changes. It's a very agile system.
That on-demand nature sounds incredibly efficient, like a bespoke message system for immediate needs. So rather than a general broadcast, it's a targeted dispatch. What kind of broad functions do they orchestrate throughout the body?
Their functions are remarkably diverse, truly touching almost every physiological system. They are deeply involved in regulating vascular tone, meaning the constriction and dilation of your blood vessels, which is critical for maintaining healthy blood pressure and directing blood flow where it's needed. They play a significant role in renal or kidney function, influencing crucial fluid and electrolyte balance. In hemostasis, the process of blood clotting, they are absolutely central. And remarkably, they are key players in parturition, the complex process of childbirth. They also maintain gastrointestinal mucosal integrity, protecting the delicate lining of your gut, and even influence the fundamental processes of stem cell function. But perhaps most crucially, and what we'll spend a good deal of time on, is their profound importance as mediators of innate immunity and inflammation, the body's very first line of defense against injury or infection.
And it's this connection to inflammation and pain that often provides our first encounter with these molecules, even if we don't realize it. And we're talking about drugs like NSAIDs, such as aspirin.
Precisely. This is one of the most direct and widely experienced therapeutic connections. Non-steroidal anti-inflammatory drugs, or NSAIDs, derive their principal therapeutic effects, whether it's relieving inflammatory pain, reducing fever, or even preventing blood clots, primarily by blocking the formation of prostaglandins, which are a major class of eicosanoids. It's a classic example of how understanding these fundamental biochemical pathways directly translates into widely used and essential medications. This isn't just theory, it's right there in your medicine cabinet.
That's a powerful entry point into understanding their significance. But how did we even begin to figure out this complex web of interactions? It sounds like an incredibly intricate system to uncover. Let's take a step back and explore the history of how these fascinating molecules were first discovered.
Yeah, the journey of discovery is truly a testament to scientific curiosity and persistence, stretching across decades. It all began back in the 1930s with some initial intriguing observations. In 1930, American gynecologists Kurzrock and Lieb, working independently, noticed something peculiar. When strips of human uterine myometrium were exposed to semen, they would either relax or contract. Around the same time, but in different countries, Goldblatt in England and Ulf von Euler in Sweden reported similar smooth muscle contracting and vasodepressor activities in seminal fluid and accessory reproductive glands.
So they knew something was present, something potent was having these effects, but they didn't quite know what it was yet. A biological mystery, basically.
Exactly. Then, building on these observations, in 1935, von Euler managed to isolate and identify the active material as a lipid-soluble acid. He named it prostaglandin, believing, somewhat incorrectly at the time, that it originated solely from the prostate gland. This foundational work by von Euler was so significant that it later contributed to him earning a Nobel Prize in 1970. The story then progresses significantly into the 1960s with the painstaking work of elucidating the precise structures and the pathways of biosynthesis.
Moving from a mysterious substance to a defined chemical structure must have been a huge leap for the field. What did that breakthrough entail?
Oh, it was monumental. In 1962, the brilliant work of Swedish scientists Sune Bergström and Bengt Samuelsson, along with their colleagues, successfully elucidated the structures of PGE1 and PGF1. This was the first time we had concrete chemical blueprints for these compounds. Then, in 1964, a major scientific breakthrough occurred when Bergström and his coworkers, along with Dutch scientist D.A. van Dorp and associates working independently, achieved the biosynthesis of PGE2 from arachidonic acid in the lab. This was absolutely crucial because it definitively showed us the primary precursor and the enzymatic pathway responsible for their creation. Following this, the discovery of other key eicosanoids like thromboxane A2 or TXA2, prostacyclin or PGI2, and the potent leukotrienes quickly followed, opening up an entirely new field of study.
And then you mentioned the therapeutic aha moment. How did aspirin, a drug that had been around for ages, fit into this newly understood biochemical picture?
That pivotal connection came in 1971. Sir John Vane, along with his colleagues Smith and Willis, reported a groundbreaking discovery. Aspirin and other NSAIDs exerted their primary effects by inhibiting prostaglandin biosynthesis. This was a direct, elegant link that explained the mechanism of action for a drug that had been in widespread use for decades, but whose precise molecular target was, unknown. It was a remarkable period of rapid discovery and profound insights, showcasing how basic science can unlock the secrets of existing medicines.
And the recognition, as you noted, was swift and well-deserved.
Absolutely. This remarkable period of intense research and breakthrough discoveries was further highlighted by another Nobel Prize in 1982, shared by Bergström, Samuelsson, and Vane. This sequence of events perfectly illustrates how a series of independent but interconnected scientific discoveries, spanning decades and involving brilliant minds from different parts of the world, can culminate in a profound understanding of an entirely new class of biological mediators, ultimately transforming both our basic knowledge of human biology and our approach to treating disease. These foundational insights truly laid the groundwork for everything we're about to delve into, providing the bedrock for mastering this field.
All right, let's dive into the actual molecules then. Section 2, eicosanoids. From precursors to products.
Let's deepen our understanding of the eicosanoids themselves, starting with their fundamental architecture. The name eicosanoids offers a direct clue to their core structure. It comes from the Greek word eikosi, which means 20.
So it's about their 20-carbon backbone, right? Like a fixed-length molecular chain.
Precisely. Eicosanoids are formed from precursor essential fatty acids that consistently contain exactly 20 carbons in their chain and either 3, 4, or 5 double bonds. These precursors are absolutely crucial because their specific structure dictates the type of eicosanoid that can be formed and consequently its biological activity. We're talking about key examples like 8-benzen-11,014-eicosanoic acid, also known as dihomolinolenic acid, the highly prevalent 5-benzen-8-benzen-11,14-eicosanoic acid, which is the infamous arachidonic acid or HOA, and 5-benzen-8-benzen-11,014,017-eicosanoic acid or EPA.
I certainly recognize arachidonic acid and EPA from discussions around nutrition and supplements. Aren't they directly related to omega-3 and omega-6 fatty acids in our diet?
You're spot on. AA is the most abundant precursor for eicosanoids in mammals, and it's derived either from the essential dietary omega-6 fatty acid, linoleic acid, or it can be ingested directly from certain foods. EPA, on the other hand, is a major constituent of oils from fatty fish like salmon, well known for their beneficial omega-3 content. This highlights a fundamental connection between our diet and the very building blocks of these powerful localized signaling molecules. And within the family of prostanoids, which are a major subtype of eicosanoids derived specifically from the cyclooxygenase pathway, numerical subscripts distinguish them based on their precursor fatty acid and the number of double bonds in their side chains. For example, prostanoids from the series 1 come from dihomolinolenic acid. Series 2, the major series in mammals, comes from ALA, and Series 3 from EPA. This numbering system gives us immediate insight into their origin and potential biological effects.
So the precursor literally sets the stage for the kind of eicosanoid that will be produced, which is an incredibly elegant and efficient system. Now, how does the body actually make these things? What's the complex process of biosynthesis that unleashes AA into action?
Right, this is where the precise orchestration of enzymes truly comes into play. The biosynthesis of eicosanoids is primarily limited by the availability of free arachidonic acid. This means that getting AA out of its stored form, typically embedded within cell membranes, is the absolutely crucial first step, acting as a control point. AA is predominantly released from esterified membrane phospholipids or other complex lipids by a group of enzymes called acyl hydrolases, most notably phospholipase A2 or PLA2. Once liberated, this free AA is then rapidly metabolized into oxygenated products by three main enzyme systems: cyclooxygenases, known as COXs, lipoxygenases or LOXs, and cytochromes P450 or CYPs.
So PLA2 is like the gatekeeper, releasing the raw material for all subsequent eicosanoid synthesis. Are there different types of PLA2, and do they work differently to fine-tune this release?
Yes, there are several important PLA2 isoforms, and their roles are quite distinct, allowing for nuanced control over AA release. Various physical, chemical, and hormonal stimuli activate these enzymes. Let's start with the group IVA cytosolic PLA2, or CPLA2. This enzyme is calcium dependent, and upon activation, it rapidly translocates from the cytosol to the cell membrane. There, it acts like a precise molecular scissor, hydrolyzing the SN2 ester bond of membrane phosphatidylcholine and phosphatidylethanolamine, which directly releases AA. This CPLA2 is particularly dominant in the acute rapid release of AA during initial stimulation, meaning it's responsible for that quick, immediate burst of eicosanoid production.
What about the other types of PLA2? Do they handle different situations, like maybe more sustained release?
Absolutely. We also have calcium-independent PLA2, or IPLA2. Under basal or normal resting conditions, the AA liberated by IPLA2 is largely reincorporated back into cell membranes through other enzymatic actions, like arachidonyl-CoA synthetase and lysophospholipid acyltransferases. This suggests IPLA2 plays a more subtle role in maintaining membrane lipid composition and perhaps a low level of basal eicosanoid signaling, rather than initiating immediate robust signaling. Then there's inducible secretory PLA2, or SPLA2. This enzyme contributes to AA release under conditions of sustained or intense stimulation, indicating its role in prolonged or heightened inflammatory responses. Interestingly, SPLA2 also contributes to the generation of eicosanoids by platelet microparticles, tiny vesicles released by platelets, which then direct the internalization of these microparticles by neutrophils, further amplifying and driving inflammation. It's a very intricate multi-level dance.
That's a fascinating level of detail, highlighting how the body finely tunes the availability of AA, the foundational building block. Once AA is liberated by these PLA2s, let's move to the next crucial stage, the products of cyclooxygenases, or COX. What are these prostanoids, and what are the critical similarities and, more importantly, the key differences between COX1 and COX2?
The cyclooxygenase pathway, colloquially known as COX, is responsible for producing a collective group of compounds we call prostanoids. These include the familiar prostaglandins, as well as prostacyclin, PGI2, and thromboxane A2, PXA2. These prostanoids are chemically distinguished by specific substitutions on their cyclopentane rings and the number of double bonds in their side chains, which is indicated by numerical subscripts. For example, prostanoids derived from arachidonic acid consistently carry the subscript 2 and constitute the major series found in mammals due to AA's abundance.
And this is where COX1 and TOX2 come in, the enzymes we hear so much about, especially when discussing anti-inflammatory medications. What are their similarities and, critically, their functional differences that make them so important in medicine?
You're right to highlight them, as they are central to both normal physiology and pharmacological intervention. COX1 and COX2 share a remarkable 61% amino acid identity, meaning their protein structures are quite similar, and they possess strikingly similar three-dimensional crystal structures. Both are glycosylated on asparagine residues, a modification that is absolutely necessary for proper protein folding and function. They're also expressed as homotypic dimers, meaning they exist as pairs of identical protein units, which are inserted into the endoplasmic reticulum membrane. Each monomer in the dimer exhibits half of the overall catalytic activity and can bind a distinct ligand that may influence the activity of the other monomer, showcasing a level of allosteric regulation. Interestingly, COX2 is also found in the Golgi apparatus and associated with lipid droplets, suggesting additional regulatory locations.
So they look and act similarly at a fundamental structural level, but their functional differences are what truly dictate their roles and the impact of drugs that target them. That's the real aha moment here, isn't it?
Indeed. This is the core insight for understanding these enzymes. COX-1 is expressed constitutively, meaning it's always present and active in most cells throughout the body. It serves as the dominant source of prostanoids for essential housekeeping functions. Think of it as your body's steady, reliable maintenance crew, producing prostaglandins crucial for processes like cytoprotection of the gastric epithelium, which safeguards your stomach lining from its own acid, or maintaining normal kidney function and platelet aggregation. In contrast, COX-2 is typically induced or upregulated dramatically by specific stimuli. These include inflammatory signals like cytokines, physical stresses like shear stress in blood vessels, and growth factors. It is the principal source of prostanoid formation during inflammatory responses, in conditions like arthritis, and importantly, in cancer. This makes COX-2 your body's crisis manager, kicking into high gear only when needed to respond to injury or disease.
That sounds like a clear division of labor, one for routine, one for emergencies. Is that distinction always so absolute, or is there some overlap?
It's a very useful conceptual distinction, but it's important to understand that this division is not entirely absolute. Both enzymes can and do contribute to autoregulatory and homeostatic prostanoid generation during various physiological and pathophysiological processes. For example, while their enzymatic properties are similar, COX-2 is actually more catalytically effective than COX-1 at lower arachidonic acid concentrations, because COX-1 exhibits negative allosterism in that setting, meaning the binding of a molecule at one site on COX-1 reduces the enzyme's activity at another site, making it less efficient when AA is scarce. Both COXs convert AA into two unstable intermediates, prostaglandin G2, PGG2, and prostaglandin H2, PGH2, through sequential cyclooxygenase and peroxidase activity. These intermediates are then rapidly converted into specific prostanoids by specialized syntheses that are expressed in a cell-specific manner. For instance, COX-1-derived TXA2 dominates in platelets, while COX-2-derived PGE2 and TXA2 dominate in activated macrophages. This cell specificity determines the final prostanoid produced.
So the cell type influences the final product, even from similar enzymes. And once these prostanoids are formed, how do they get out of the cell to do their job, and how does the body clear them?
Right. Prostanoids are generally released from cells by simple diffusion across the cell membrane, though their transport can be facilitated by specific protein transporters, such as the multi-drug resistance-associated protein transporter. There's also a dedicated prostaglandin transporter, or PGT, which specifically transports extracellular prostaglandins into the cytoplasm for degradation, highlighting a crucial mechanism for their clearance and precise regulation. Furthermore, while COX-1 and COX-2 are structurally similar, COX-2 has a broader substrate preference. This means it can oxygenate not just AA, but also neutral derivatives of AA and other fatty acids, giving it more versatility. There's also variable functional complementarity between them. In the kidney, for example, if COX-2 is lost, COX-1 cannot compensate. But if COX-1 is lost, COX-2 can compensate. Similarly, in the female reproductive system, COX-1 function may be replaced by COX-2, but not vice versa. This indicates a sophisticated built-in backup system in certain tissues, adding another layer of complexity to their roles.
That's incredible built-in redundancy and specificity, depending on the tissue. Now, beyond the COX pathway, you mentioned lipoxygenases or LOXs. What do these enzymes produce, and how do they contribute to the body's intricate signaling, particularly in inflammation?
The LOX pathways primarily produce hydroxy fatty acid derivatives, known as HEAS, as well as the potent leukotrienes, or LTs, and lipoxins, or LXs. Leukotrienes in particular play a major and often detrimental role in the development and persistence of the inflammatory response, especially in conditions like asthma. LOXs themselves are a family of enzymes containing non-heme iron. They catalyze the precise oxygenation of polyenic fatty acids, specifically those with two cis double bonds separated by a methylene group, a structural feature that arachidonic acid possesses into corresponding lipid hydroperoxides, known as HPEs. These HPTs are then converted to their corresponding heats, either non-enzymatically or by a peroxidase enzyme.
Are there different types of LOXs similar to the COX enzymes with specific tissue distributions?
Yes, there are 5 active human LOXs, each classified by the specific site on the fatty acid chain where they insert the hydroperoxy group: 5S LOX, 12S LOX, 12R LOX, 15S LOX1, and 15S LOX2. Their expression is often highly cell-specific, indicating specialized functions. For example, platelets primarily have only 12S LOX, while various leukocytes, your white blood cells, contain both 5S and 12S LOX. Interestingly, 12 LOX is mostly restricted to the skin. Epidermal LOXs also include 15 LOX2 and ELOX3, with ELOX3 uniquely metabolizing 12R heat to a specific epoxy-alkyl product, showing a very specific biochemical role in the skin.
And the 5-LOX pathway sounds particularly important because it leads directly to the formation of leukotrienes, which are major players in inflammation. Can you walk us through that pathway?
It absolutely does. The 5-LOX pathway is critical for the synthesis of all leukotrienes. When cells like eosinophils, mast cells, polymorphonuclear leukocytes, PMNs, or monocytes are activated, the 5-LOX enzyme translocates from the cytosol to the nuclear membrane. There it forms a crucial association with 5-LOX activating protein, or FLAP, an integral membrane protein that acts as a chaperone, facilitating the interaction between arachidonic acid and 5-LOX. This tight association is why drugs that inhibit FLAP are effective at blocking leukotriene production. 5-LOX then catalyzes a two-step reaction. First, the oxygenation of AA to form 5-HPEAT, which is then immediately followed by a dehydration step to produce an unstable epoxide, leukotriene A4 or LTA4. LTA4 is a crucial intermediate because it can then be transformed by distinct specific enzymes into two primary products. LTB4, a potent chemoattractant, or LTC4. Further extracellular metabolism of LTC4's peptide moiety generates LTD4 and LTE4. Collectively, LTC4, LTD4, and LTE4 are known as the cysteinyl leukotrienes, or cis-LOTs, due to their cysteine-containing structure. Both LTB4 and LTC4 are actively transported out of the cell to exert their potent inflammatory effects.
And what about lipoxins? You mentioned those earlier in this pathway. Are they just byproducts or do they have a role?
Yes, LTA4 can also be metabolized by 12 LOX to form lipoxin A4, LXA4, and lipoxin B4, LXB4. These mediators can also arise from 5-LOX metabolism of 15 heat. It's worth noting that while they show promise in research, the precise biological relevance of lipoxins as endogenous active mediators in vivo remains a controversial topic among scientists. However, LXA4 can activate a specific G protein-coupled receptor that is also activated by annexin A1 and formyl peptide, known as the ANX-A1 formyl peptide receptor 2-ALX receptor, suggesting a potential role in anti-inflammation or resolution.
So many pathways, so many products, all from a single precursor. What about the third enzyme system you mentioned, the cytochromes P450 or CYPs? What unique products do they produce and what role do they play?
The CYP epoxygenases, primarily specific isoforms like CYP2C and CYP2J, metabolize arachidonic acid to epoxyeicosanoic acids, or EETs. These molecules were initially described in endothelial cells, the cells lining your blood vessels, where they function as endothelin-derived hyperpolarizing factors, or EDHFs. This means they promote vasodilation, especially in crucial areas like the coronary circulation, helping blood vessels relax and widen, improving blood flow.
That sounds like a protective role, helping to ensure healthy blood flow. Are they only found in endothelial cells, or are they more widespread?
No, ETs can actually be produced by any cell expressing CYP epoxygenase, making them quite ubiquitous. This includes cells like astrocytes in the brain, and they may play a significant protective role in cerebral ischemia, a condition where blood flow to the brain is reduced, potentially limiting damage. ET biosynthesis can be altered by various factors, including pharmacological interventions, specific nutritional choices, and even inherited genetic factors that affect CYP expression, giving us potential avenues for modulating their levels and effects.
Beyond these main three enzymatic pathways, COX, LOX, and CYPs, are there any other noteworthy or emerging pathways for AA-derived products that contribute to this complex lipid language?
Yes, there are other intriguing and important pathways that add to the complexity of this metabolic network. One involves isoeicosanoids, which are a family of eicosanoid isomers. Unlike the enzymatically formed prostaglandins, these compounds are generated by non-enzymatic, free radical catalyzed oxidation of arachidonic acid, often occurring during oxidative stress. A unique aspect is that they are initially formed, esterified within phospholipids in cell membranes, and are then subsequently released by phospholipases. Once released, they circulate throughout the body, are metabolized, and eventually excreted into the urine. Their production is notably not inhibited by COX-1 or COX-2 inhibitors, setting them apart, but it is suppressed by antioxidants. Isoeicosanoids correlate with cardiovascular risk factors, and increased levels are found in many clinical conditions, but their precise relevance as biologically active mediators in their own right remains somewhat speculative and is an active area of research.
And what about specialized pro-resolving mediators? That sounds like a crucial step for turning off inflammation once it's done its job.
Indeed. These include compounds like the lipoxins we mentioned, as well as myrcins and resolvins. When synthesized and administered to certain models of inflammation, they demonstrate a remarkable ability to hasten its resolution, effectively helping the body return to a normal, non-inflamed state after an immune response. However, it remains a key open question in the field whether these endogenous compounds are formed in sufficient quantities in vivo within our bodies to exert this full effect under natural conditions. Finally, it's absolutely crucial to understand the metabolic network concept. Since COX, LOX, and CYP products are all formed from arachidonic acid, their biosynthesis should not be viewed in isolation. They are part of the same interconnected metabolic network. Therefore, comprehensive assessment often requires broad-spectrum eicosanoid assays to fully uncover this intricate interconnectivity and any potential substrate rediversion, where blocking one pathway might push AA towards another in response to various physiological or pharmacological perturbations. It's a holistic system, not just a series of independent reactions.
Right, that makes sense. You can't just look at one piece. Okay, section 3. Regulating eicosanoid action inhibition and degradation. We've explored how they're synthesized, detailing the intricate pathways. Now, let's turn our attention to how their actions are controlled, specifically through inhibition of their production and their eventual degradation. This is where we often see the direct intersection with pharmaceuticals and therapeutic strategies. Let's begin with inhibitors of eicosanoid biosynthesis.
This is a truly critical area for therapeutics, as it's how we manipulate these powerful signals. Inhibiting PLA2, the enzyme we discussed earlier that's responsible for releasing arachidonic acid from membranes, will directly decrease the availability of this precursor fatty acid. Consequently, this will reduce the synthesis of all its downstream metabolites, prostaglandins, leukotrienes, and ETs. One way to inhibit PLA2 is by using drugs that reduce the availability of intracellular calcium, as many PLA2 isoforms are calcium dependent. Interestingly, glucocorticoids, a widely used class of steroid hormones, inhibit PLA2 indirectly. They do this by inducing a group of proteins called annexins, which modulate PLA2 activity. Beyond this, glucocorticoids also have a broader effect of downregulating the induced expression of COX2, but they specifically do not affect the constitutive COX1 expression, contributing to their potent anti-inflammatory properties without disrupting essential COX1 functions.
And this brings us back to the most common type of inhibitors, the COX inhibitors, the NSAID story. How do they fit into this inhibition mechanism, and what are the crucial implications of their action, especially with that idea of substrate shunting you mentioned?
Right. Aspirin and the vast majority of non-selective NSAIDs primarily exert their therapeutic effects by inhibiting the cyclooxygenase activity, but not the peroxidase activity, of both COX1 and COX2 enzymes. This effectively blocks the formation of downstream prostanoids. A crucial and sometimes problematic point here is what we call substrate shunting. Think of it like a major highway with two main exits. If you block one main exit, all the traffic that was heading there will suddenly divert to the other, often less suitable, exit. In our bodies, when NSAIDs block the COX pathway, the raw material arachidonic acid doesn't just disappear. Instead, it gets shunted towards the LOX pathway because that pathway is still open. This can cause an increased formation of leukotrienes. This shunting mechanism is a significant factor contributing to some of the adverse gastrointestinal effects often associated with traditional NSAIDs, such as stomach upset and ulcers, because leukotrienes can be pro-inflammatory in the gut.
That's a classic example of unintended consequences in pharmacology, showing how complex these pathways are. Did this understanding of shunting and the distinct roles of COX1 and COX2 directly lead to the development of more specific inhibitors?
It absolutely did. The understanding of this shunting mechanism led to the development of dual inhibitors of COX and 5-LOX. These drugs have shown effectiveness in some models of inflammation and tissue injury, and importantly, may lessen the degree of adverse GI effects compared to traditional COX inhibitors by preventing that problematic substrate diversion. Furthermore, the perceived differences in sensitivity of COX1 and COX2 to anti-inflammatory drugs, along with their distinct physiological roles, paved the way for the development of selective COX2 inhibitors, commonly known as COXs.
Ah, the COXs. They were initially hailed as a major breakthrough, specifically because they promised better GI safety, right? That was the big selling point.
That was indeed the compelling rationale. These drugs were hypothesized to offer significant therapeutic advantages over older, non-selective NSAIDs because COX2 was believed to be the predominant source of prostaglandins in inflammation and pain, while COX1 was considered the major source of cytoprotective prostaglandins in the GI tract and platelet TXA2, important for clotting. And initially, randomized clinical trials did report their superiority in GI safety compared to non-selective NSAID comparators, validating that aspect of the hypothesis.
But then we learned about the cardiovascular hazards. That was a major curveball, a truly unexpected and serious consequence for the medical community and patients. How do we explain that now?
It was indeed a significant unforeseen challenge that fundamentally reshaped our understanding. Compelling evidence emerged demonstrating that COX-2 inhibitors confer a spectrum of serious cardiovascular hazards. These include an increased risk of myocardial infarction or heart attack, stroke, systemic and pulmonary hypertension, congestive heart failure, and even sudden cardiac death. The explanation for these hazards is now sufficiently clear and highlights the delicate balance of prostanoids. These adverse effects are largely explained by the suppression of cardioprotective COX-2-derived prostaglandins, especially prostacyclin, or PGA2. This suppression, in turn, is coupled with the unrestrained effects of endogenous stimuli, such as platelet, COX-1-derived TXA2, on platelet activation, vascular proliferation, remodeling, hypertension, and atherogenesis. Essentially, while they inhibited the bad inflammatory COX-2, they also inhibited the good cardioprotective COX-2, which normally provides a crucial brake on clotting and vascular constriction, allowing unrestrained activity from the good housekeeper COX-1, which in platelets means more clotting. It effectively removed a natural protective balance.
So targeting one pathway had a profound domino effect on another, revealing a much more complex and critical interplay than initially understood.
Wow. Now, what about the leukotriene pathway? We've seen its significant role in inflammation. How do we target it therapeutically, for example, in conditions like asthma?
Given that leukotrienes are potent and often problematic mediators of inflammation, significant efforts have focused on developing drugs that target this pathway. These include leukotriene receptor antagonists, which block the action of leukotrienes at their receptors, and selective LOX inhibitors, which prevent their formation. Key drugs in this category include Xiliton, which is an inhibitor of 5-LOX, blocking the initial synthesis of all leukotrienes. Then there are the selective cis-LTR1 antagonists, such as efferleucas, prenleucas, and monteleucas, which specifically block the receptors for sustainol leukotrienes. These drugs have established clinical efficacy in the treatment of mild to moderate asthma, although they generally remain less effective than inhaled corticosteroids, which act more broadly on inflammation. And are there genetic factors that influence how effective these treatments are, or how an individual might respond to them? That sounds like the cutting edge of personalized medicine.
Yes. Genetics absolutely play a role, pointing towards future possibilities for personalized treatment. A common polymorphism, or variation, in the gene for LTC4 synthase, the enzyme that forms LTC4, correlates with increased LTC4 generation. This genetic variation may be associated with a higher asthma risk in some populations and can also influence the efficacy of anti-leukotriene therapy. Smaller studies have also reported that polymorphisms in the genes encoding 5-LOX or FLAP may be linked not only to asthma and lung function, but also to myocardial infarction, stroke, and atherosclerosis. This growing body of evidence suggests that inhibiting leukotriene biosynthesis may eventually prove useful not just for asthma, but potentially in the prevention of cardiovascular disease, opening up exciting new therapeutic avenues beyond just inflammatory conditions.
Fascinating. So, beyond inhibiting their production, how does the body naturally get rid of eicosanoids once they've done their job? What does the cleanup crew look like for these potent transient molecules?
Eicosanoids are efficiently and rapidly inactivated. This rapid breakdown is a key reason for their localized and transient nature. They act quickly and then disappear, preventing widespread or prolonged effects. The enzymatic catabolic reactions generally occur in two types of steps. The first is a rapid initial step, catalyzed by widely distributed prostaglandin-specific enzymes. In this crucial first pass, prostaglandins lose most of their biological activity. Following this, there's a second step where these initial metabolites are further oxidized, likely by enzymes identical to those responsible for the beta and omega oxidation of fatty acids, effectively dismantling them completely for excretion.
Which organs are primarily responsible for this metabolic clearance, acting as the major processing centers?
The lung, kidney, and liver play prominent roles in these enzymatically catalyzed reactions, acting as major sites of eicosanoid metabolism and clearance. This metabolic clearance requires an energy-dependent cellular uptake, primarily by the prostaglandin transporter, or PGT, and possibly other transporters, which facilitates their movement into the cells for degradation. The crucial initial enzymatic step for many prostaglandins is the oxidation of the 15-OH group to the corresponding ketone by 15-hydroxyprostaglandin dehydrogenase, or 15-PGDH. This is often the first and most important step in their inactivation. However, there are important exceptions. PGI2 and TXA2, for instance, undergo spontaneous hydrolysis as their first degradative step, meaning they break down on their own simply by reacting with water without needing an enzyme to initiate the process. This contributes to their extremely short half-lives.
And how is LTC4 specifically degraded, which is so relevant for lung issues like asthma?
LTC4 degradation occurs in the lungs, kidney, and liver. but it may also happen in myeloid tissue via specific CYP4F enzymes. The clinical relevance of the 15-PGDH enzyme we just mentioned is quite interesting and multifaceted. Inactivation of 15-PGDH, either pharmacologically or genetically, elevates the capacity of tissues to form PGE2. This enhanced PGE2 level can then enhance tissue regeneration after hematopoietic stem cell transplantation and even accelerate recovery after hemihepatectomy, which is the surgical removal of part of the liver. This points to a beneficial role of PGD2 in tissue repair. On the flip side, its enhanced expression with advancing age has been speculated to contribute to sarcopenia, which is age-related muscle loss, suggesting that too much 15-PGDH might contribute to chronic conditions of aging by inappropriately clearing beneficial prostaglandins. This enzyme clearly has a dual context-dependent role.
Okay. Section 4. Mechanisms of action. Receptors and signaling. We've gone deep into how eicosanoids are produced and degraded. Now, how do they actually exert their effects? How do these molecules communicate their message to the cell? Is it like a lock-and-key system?
You've hit on a perfect analogy. Eicosanoids primarily exert their functions by activating specific G-protein-coupled receptors, or GPCRs, which are specialized proteins embedded in the cell surface membrane. Think of these GPCRs as the locks, and the eicosanoids as the very specific keys. When the right eicosanoid key binds to its GPCR lock, it causes a conformational change that then activates a G protein inside the cell. These G proteins then couple to various intracellular second messenger systems, such as adenylyl cyclase, which modulates cyclic AMP levels, and phospholipase C, or PLC, which affects calcium signaling. This then sets off a cascade of events, much like a series of dominoes falling, to modulate various cellular activities. It's a classic signaling pathway where an external signal is amplified and precisely translated into an internal cellular response.
So each eicosanoid type must have its own specific receptors to ensure the right message gets to the right place, preventing miscommunication. Tell us more about the prostaglandin receptors. How diverse are they, and what different signals do they initiate?
Right. Prostaglandins predominantly activate these membrane receptors locally, very close to their sites of formation, which really underscores their paracrine or local signaling nature, as opposed to acting like hormones that travel widely throughout the bloodstream. Eicosanoid receptors interact with different types of G proteins, Gs, G, and GQ, which in turn modulate the activities of adenylyl cyclase, leading to changes in cyclic AMP levels and PLC affecting intracellular calcium. This diversity in G protein coupling is a major reason for the diverse cellular responses we see.
And how diverse are these receptors themselves? Is it one receptor per prostaglandin, or is it much more intricate than that?
Oh, it's remarkably intricate. While single gene products have been identified for the receptors for PGI2, known as the ITR, PGF2, the FPR, and TXA2, the TPR, it gets far more complex with PGE2 and PTD2. For PGE2, 4 distinct receptors have been cloned: EPR1, EPR2, EPR3, and EPR4. For PGD2, there are 2 receptors, DPR1 and DPR2, with DPR2 also known as CRTH2. What's more, additional isoforms of the TPR, FPR, and EPR3 receptors can arise through differential messenger RNA splicing, a process where different protein versions are made from the same gene, allowing for even greater diversity in their signaling capabilities within different cell types.
So a single prostaglandin, like PGE2, can potentially bind to multiple receptor types, leading to completely different or even opposing effects depending on which receptor is activated. That's a truly sophisticated system.
Precisely. This receptor diversity allows for very fine-tuned and context-dependent actions, ensuring that the same molecule can trigger different responses in different cells or under different conditions. When we look at the prostanoid receptors phylogenetically, meaning their evolutionary relationships, they appear to derive from an ancestral EPR and share high homology or structural similarity. This allows us to group them into 3 broad functional subclusters. First, we have the relaxant receptors, EPR2, EPR4, IPR, and DPR1. These primarily increase cellular cyclic AMP generation, which often leads to the relaxation of smooth muscle cells or the inhibition of certain cell functions. Second, there are the contractile receptors, EPR1, FPR, and TPR. These primarily increase cytosolic levels of calcium, which often triggers contraction, secretion, or activation of cells. And then EPR3 stands out as unique, as it can couple to both the elevation of cytosolic calcium and the inhibition of adenylyl cyclase, providing a remarkably versatile signaling profile that can switch between different outcomes.
Is the DPR2 receptor an exception to this tidy clustering, or does it fit in somehow?
Yes. The DPR2 receptor is indeed an exception to this specific phylogenetic clustering. It is unrelated to the other prostanoid receptors and is instead a member of the FMLP, or formal methynyl leucylphenylalanine receptor superfamily. This highlights the varied evolutionary origins of these signaling components, showing that similar functions can arise from different genetic lineages. Our understanding of these receptors has recently advanced significantly with the resolution of the crystal structures of the active and inactive states of various human prostaglandin receptors, including DPR2, EPR3, EPR4, and PPR. These structural insights are critically important for rational drug development, allowing scientists to design molecules that fit precisely into these receptor pockets, leading to more targeted and effective therapies.
That's a huge step for targeted therapies, truly enabling precision medicine. What about the leukotriene receptors? How do they mediate the signals that drive inflammation?
For LTB4, a potent chemoattractant, two distinct receptors exist, BLT1 and BLT2. For the cisstanol leukotrienes, or CYPs, LTC4, LTD4, and LTE4, there are three identified receptors: CYPLTR1, CYPLTR2, and CYPLTR3, with CYPLTR3 also known as OXGR1 or GPR99. Interestingly, the FMLP2 receptor, which is part of the same superfamily as DPR2, also binds LXA4, a lipoxin, though the precise functional importance of this ligand in vivo still remains a subject of ongoing research. All leukotriene receptors are also GPCRs and couple with GQ and other G proteins depending on the specific cellular context and stimulus.
And how are these leukotriene receptors distributed throughout the body, and what are their specific affinities for the different leukotrienes? This must be key to understanding their highly localized inflammatory effects.
Their distribution and binding affinities are absolutely crucial for their specific effects in inflammatory and immune responses. BLT1 is predominantly expressed in leukocytes, the thymus, and the spleen, indicating its primary role in immune cell function and recruitment. BLT2 is the lower affinity receptor for LTB4 and is found more broadly in the spleen, other leukocytes, the ovary, liver, and intestine, suggesting a more widespread, perhaps homeostatic role. For CYPLTs, CYPLTR1 binds LTD4 with a higher affinity than LTC4, making it a key target for LTD4's potent bronchoconstrictor effects. CYPLTR2, on the other hand, shows equal affinity for both LTC4 and LTD4. Both CYPLTR1 and CYPLTR2 bind LTE4 of relatively low affinity. However, CYPLTR3 appears to have high affinity only for LTE4, indicating a very specific role for this particular leukotriene. In terms of signaling, activation of GQ, leading to the mobilization of intracellular calcium, is the primary signaling pathway reported for these receptors, although studies also place GI downstream of CYPLTR2, again showing a nuanced and adaptable signaling profile.
That cell and tissue specificity is clearly key for understanding their physiological and pathological actions. Where do we find these CYPLT receptors primarily expressed?
CYPLTR1 is expressed in lung and intestinal smooth muscle, the spleen, and peripheral blood leukocytes, indicating its role in respiratory and gastrointestinal responses, as well as immune cell function. CYPLTR2 is found in the heart, spleen, peripheral blood leukocytes, adrenal medulla, and brain, suggesting broader systemic involvement. CYPLTR3 is expressed in the kidney, trachea, lung, salivary glands, airway epithelial cells, and mast cells, reinforcing its role in allergic and respiratory conditions. Like the prostaglandin receptors, the recent groundbreaking resolution of the crystal structures for CYPLTR1, CYPLTR2, and BLT1 is a monumental step forward. These structural insights could significantly facilitate the development of highly specific and effective new therapeutic agents targeting these receptors, offering more precise tools to modulate inflammatory responses in diseases like asthma and allergies.
It seems like understanding these receptors down to their atomic structure is fundamental to designing precision drugs. Are there any other emerging players or receptors for other less discussed arachidonic acid-derived products?
Yes, indeed. The field is constantly expanding. Other arachidonic acid-derived products, such as isoprosanes and the ETs we discussed, also exhibit potent biological activities, and there is growing evidence for distinct receptors for some of these substances, hinting at an even more complex signaling landscape. For example, an orphan receptor named GPR31 has been identified as a receptor for 12-SATE, and recent evidence suggests that another orphan receptor, GPR75, functions as a receptor for 20-ATE. The continued discovery and characterization of these receptors reveal the vast and intricate network of lipid signaling within the body, suggesting that we are still uncovering the full lipid language our cells speak.
Okay, Section 5. Physiological and pathological actions eicosanoids across systems. Here's where it gets really interesting, and the sheer scope of these molecules becomes apparent. With so many different eicosanoid types, diverse synthetic pathways, and multiple receptors, you can imagine the sheer complexity of their roles. It's like a sophisticated symphony of signals happening all over your body, impacting virtually every system. What does this all mean for us? How do these intricate molecular details translate into physiological effects, both healthy and pathological?
Indeed. The widespread biosynthesis and myriad pharmacological actions of eicosanoids are truly reflected in their complex physiology and pathophysiology. Mastering this topic requires understanding not just the molecules, but how their presence and absence orchestrate changes throughout the organism. Understanding the distribution of their major receptors across different tissues helps put this complexity into perspective. Furthermore, studies using mice with targeted disruptions of genes regulating eicosanoid biosynthesis and receptors have been invaluable. These knockout studies have unveiled unexpected roles and clarified long-standing hypotheses about their functions, providing critical insights into how these tiny molecules orchestrate massive changes in health and disease. Let's break down some of their most significant impacts across the body, starting with the cardiovascular system.
Let's do it. So how do these short-lived molecules regulate something as fundamental as blood flow and vascular tone, considering they don't circulate widely like hormones?
Because of their very short half-lives, typically just seconds to minutes at physiological pH, the effects of endogenous prostanoids on vascular tone primarily occur locally at their sites of biosynthesis. They act as paracrine mediators, meaning they signal to nearby cells rather than traveling throughout the bloodstream to exert systemic effects. Any systemic effects of prostanoids on blood pressure usually involve their secondary influence on renal function and hormonal processes.
So localized vasodilation, the widening of blood vessels, is a key function. Which eicosanoids are involved in that, and how do they achieve it?
Prostacyclin, or PGI2, is a prime example. It's a major arachidonic metabolite released from the vascular endothelium, primarily derived from COX2 in humans. PGI2 is widely considered atheroprotective, meaning it helps prevent the hardening and narrowing of arteries, a key factor in heart disease. Its generation and release are finely regulated by mechanical forces like shear stress, the friction of blood flow against vessel walls, and by both vasoconstrictor and vasodilator autacoids, demonstrating a complex feedback loop. In most vascular beds, PGE2, PGI2, and PGD2 are the primary prostanoids that elicit vasodilation. While infusing high concentrations of these systemically can cause a drop in blood pressure, physiologically, these responses are quite localized due to their rapid inactivation. Then we have the epoxyeicosantranoic acids, or EETs, produced by CYP epoxygenases. These cause vasodilation in several vascular beds by activating large conductance calcium-activated potassium channels in smooth muscle cells, leading to hyperpolarization and relaxation. ETs also function as endothelium-derived hyperpolarizing factors, particularly in the coronary circulation. Interestingly, endogenous biosynthesis of ETs is increased in human syndromes of hypertension, suggesting a complex, perhaps compensatory or even pathological role in blood pressure regulation that we're still working to fully understand.
And on the flip side, what about vasoconstriction, the tightening of blood vessels? Which eicosanoids are responsible for that, and in what contexts?
PGF2 is a potent constrictor of both pulmonary arteries and veins, playing a role in regulating blood flow through the lungs. Thromboxane A2, or TXA2, is a very potent vasoconstrictor and also acts as a mitogen, meaning it stimulates cell proliferation in smooth muscle cells, which can contribute to vascular remodeling and disease. PGE2, surprisingly, can also cause vasoconstriction, specifically through activation of its EP1 and EP3 receptors, highlighting its dual nature depending on the receptor subtype. PGD2, when infused systemically in humans, can result in flushing, nasal congestion, and hypotension. Its local subcutaneous release contributes to the dilation of skin vasculature, causing the facial flushing often associated with niacin treatment. However, subsequent formation of efferine metabolites from PGD2 may paradoxically result in hypertension, demonstrating complex metabolic shifts.
And leukotrienes also have a significant impact on blood vessels, correct? Not just prostaglandins.
Yes. LTC4 and LTD4 can either constrict or relax isolated vascular smooth muscle preparations, depending on the concentrations and the specific vascular bed being studied. While they have little effect on most large arteries or veins, even nanomolar concentrations, very low amounts, can potently contract coronary arteries and distal pulmonary artery segments, making them significant in certain circulatory areas. The renal vasculature is notably resistant to this constrictor action, but the mesenteric vasculature is not, showing tissue-specific sensitivity. These leukotrienes are also incredibly potent in increasing vascular permeability. LTC4 and LTD4 act in the microvasculature to dramatically increase the permeability of postcapillary venules. They are about 1,000-fold more potent than histamine in this regard, leading to rapid fluid leakage and edema at inflammatory sites. At higher concentrations, they can also constrict arterioles and reduce the exudation of plasma, demonstrating a concentration-dependent effect.
Beyond flow and tone, how do eicosanoids affect cardiac output and heart protection? Do they contribute to heart health or disease?
Well, systemic infusion of prostaglandins of the E and F series generally increases cardiac output, while weak, direct inotropic effects, meaning affecting the contractile force of heart muscle, have been noted in isolated preparations. In intact animals, increased force and heart rate are largely a reflex consequence of a fall in total peripheral resistance induced by their vasodilatory effects. More importantly, PGA2 and PGE2, acting on the IPR or the EPR3 respectively, play critical roles in protecting against oxidative injury in cardiac tissue. We've seen that IPR deletion in animal models significantly augments myocardial ischemia or perfusion injury damage that occurs when blood flow returns to oxygen-deprived heart tissue. Similarly, both microsomal PGE synthase 1 deletion and cardiomyocyte-specific deletion of the EPR4 exacerbate the decline in cardiac function after experimental myocardial infarction. This highlights their crucial protective roles in heart function and recovery.
So they have clear protective roles, but can these same molecules also be harmful to the heart? It seems that there's always a balance.
They absolutely can, highlighting that delicate balance. For instance, COX2-derived TXI2 has been shown to contribute to oxidant stress, isoprostane generation, and activation of the TPR, and possibly the FPR, leading to increased cardiomyocyte apoptosis, programmed cell death, and fibrosis, which is scarring in a model of heart failure. And in mice, selective deletion of COX2 specifically in cardiomyocytes actually results in mild heart failure and a predisposition to arrhythmogenesis, or abnormal heart rhythms, demonstrating that even the good KROX2 can have nuanced or detrimental roles if its balance is disturbed. Finally, there is compelling evidence for a significant role of leukotrienes in cardiovascular disease. Human genetic studies have even indicated a link between cardiovascular disease and specific polymorphisms in the genes encoding the leukotriene biosynthetic enzymes and FLAP, suggesting that individual genetic variations can predispose to cardiovascular risk via these pathways. It's a truly complex and often contradictory picture that requires a nuanced understanding.
That's a lot of intricate regulation within the cardiovascular system. Let's move to a very specific, crucial perinatal role, the patent ductus arteriosus. How do eicosanoids govern the opening and closing of this vital fetal structure?
This is a truly beautiful and precise example of eicosanoid control that is literally a matter of life or death at birth. During fetal development, cyclooxygenase 2-derived PGE2, acting primarily via the EPR4, maintains the ductus arteriosus patent, or open. This allows blood to bypass the developing lungs, which are not yet functional. After birth, a natural reduction in PGE2 levels, a consequence of increased PGE2 metabolism and changes in oxygen tension, then permits its crucial closure, redirecting blood flow to the lungs. This is why traditional NSAIDs, by inhibiting PGE2 formation, can be used clinically to induce closure of a patent ductus in neonates who might otherwise struggle to breathe. Strikingly, animal studies underscore this mechanism. Mice lacking the EPR4 during development actually die with a patent ductus during the perinatal period because the precise mechanism for control of the ductus in utero and its essential remodeling at birth is absent without this specific receptor. It's a direct, profound illustration of their importance.
That's a powerful life or death example of their importance. Now let's talk about platelets, those tiny cell fragments crucial for blood clotting. How do eicosanoids navigate the delicate balance between promoting clotting and preventing excessive bleeding?
Platelet aggregation is a process fundamental to hemostasis, and it leads to the activation of membrane phospholipases, releasing AA and subsequent eicosanoid biosynthesis. In human platelets, TXA2 and 12-HT are the two major eicosanoids formed, though eicosanoids from other sources, like PGI2 from vascular endothelium, also significantly affect platelet function. A critical point for understanding therapeutic intervention is that mature platelets express only COX1. They do not have COX2. TXA2, which is the major product of COX1 in platelets, is a potent inducer of platelet aggregation and powerfully amplifies the signal for other more potent platelet agonists such as thrombin and ADP, essentially pushing the clotting button.
So TXA2 is the key pro-clotting force within platelets, and it comes solely from COX1. And this precisely explains why low-dose aspirin, by targeting COX1, is so important for cardiovascular prevention, right?
Exactly. The immense importance of the TXA2 pathway is clearly evident from the established efficacy of platelet COX1 inhibition with low-dose aspirin in the secondary prevention of myocardial infarction and ischemic stroke. Aspirin irreversibly inhibits platelet COX1, reducing TXA2 synthesis for the lifespan of the platelet, thereby reducing unwanted clotting. We can even gauge the overall platelet activation in vivo by measuring the total biosynthesis of TXA2, as determined by the excretion of its urinary metabolites. This is often augmented in clinical syndromes of platelet activation, including unstable angina, myocardial infarction, and stroke, serving as a valuable biomarker. Genetic insights also highlight its importance. Deletion of the TPR, the TXA2 receptor, in mice prolongs bleeding time, renders platelets unresponsive to TPR agonists, and blunts the response to vasopressors and the proliferative response to vascular injury, underscoring its multifaceted role. In terms of signaling, TXA2 induces platelet shape change through G12-G13-mediated RORO kinase-dependent regulation of myosin light chain phosphorylation, and it drives aggregation through GQ-dependent activation of protein kinase C, illustrating precise intracellular pathways.
So, TXA2 pushes clotting aggressively. What keeps it in check? What are the natural brakes on platelet activation?
The powerful actions of TXA2 on platelets are naturally restrained by several mechanisms. First, its very short half-life, around 30 seconds, means its effects are transient. Second, there's rapid TPR desensitization, where the receptor quickly becomes less responsive after activation. And crucially, there are powerful endogenous inhibitors of platelet function, including nitric oxide, NO, which is a vasodilator and antiplatelet agent, and PGI2, or prostacyclin, which is also produced by vascular endothelial cells. This complex balance is absolutely crucial for preventing both excessive bleeding and unwanted clotting. Interestingly, low concentrations of PGE2 via the EPR3 receptor can actually enhance platelet aggregation. In contrast, higher concentrations of PGE2, acting via the IPR or possibly EPR2 or EPR4, inhibit platelet aggregation, showcasing its dual concentration-dependent role. Both PGI2 and PGD2 also inhibit the aggregation of platelets, with PGI2 specifically limiting platelet activation by TXA2 and even able to disaggregate preformed platelet clumps, acting as a direct counterbalance.
And this brings us back, full circle, to the cardiovascular hazards of selective COX2 inhibitors. This is the crucial point for understanding those risks.
Precisely. The increased incidence of myocardial infarction and stroke in patients receiving selective inhibitors of COX2 is a direct consequence of the inhibition of COX2-dependent PGI2 formation. Remember, PGI2 is the cardioprotective molecule that normally acts as a brake on platelet activation and promotes vasodilation. By selectively inhibiting COX2, these drugs remove this crucial brake on platelet activation while leaving the powerful COX1-derived TXA2 activity in platelets unrestrained. This imbalance shifts the system towards a prothrombotic state, increasing the risk of clots that can lead to heart attacks and strokes. It's a stark and vital reminder of the delicate and often unpredictable balance these eicosanoids maintain in our physiology.
That's a complex but absolutely crucial system to understand for anyone interested in pharmacology and cardiovascular health. Let's shift gears to inflammation and immunity, where eicosanoids play an equally central and dynamic role in orchestrating the body's defense mechanisms.
Absolutely. Eicosanoids are major, multifaceted players in inflammatory and immune responses. Leukotrienes generally are profoundly pro-inflammatory and interact synergistically with prostaglandins to promote and sustain inflammation. However, there are some important exceptions, such as the inhibitory actions of PGE2 on mast cell activation, highlighting their nuanced and sometimes contradictory roles depending on the context. Prostaglandins and lipoxins, along with related specialized pro-resolving lipid mediators, may also contribute actively to the resolution of inflammation, helping the body return to a state of balance and healing after an immune response. COX2 is unequivocally the major source of prostanoids formed during and after an inflammatory response, emphasizing its role in acute inflammation and its progression.
This sounds incredibly relevant to what we've experienced recently with global health crises, particularly viral infections. How do eicosanoids play a role in, say, SARS-CoV-2 infection and the pathology of COVID-19?
The role of eicosanoids in viral infections, particularly SARS-CoV-2, is a fascinating and critical area. Eicosanoids can profoundly modulate virus infection by regulating complex virus-host interactions and shaping the host immune and inflammatory responses activated by viral pathogens. For instance, specific eicosanoids may directly influence SARS-CoV-2 entry to cells, its replication within the cell, and the eventual clearance of the virus by the immune system. Research has even suggested that the non-structural SARS-CoV-2 protein NSP7 may interact with microsomal prostaglandin E synthase 2, a key enzyme in PGE2 synthesis, implying a direct viral manipulation of eicosanoid pathways. We also know that infection by various viruses can broadly impact the expression of genes for proteins in the arachidonic acid pathway and the biosynthesis of bioactive lipids, demonstrating a dynamic interplay where the virus can hijack or alter host lipid signaling.
So specific eicosanoids might directly contribute to the severe cytokine storm and hyperinflammation we've observed in critically ill COVID-19 patients.
Precisely. PGE2 through its complex effects on type I interferon signaling, cytotoxic T-cell and Th17 responses, macrophage polarization, natural killer cell functions, neutrophil extracellular trap formation, inflammasome activation, and nuclear factor key EB activation, all key immune pathways, may contribute significantly to the exaggerated immune response and cytokine storm observed in severe COVID-19 patients. Similarly, CYPs may profoundly regulate the immune and inflammatory response to SARS-CoV-2 infection by having a potent chemoattractant effect on neutrophils and lymphocytes and by causing macrophage activation and pro-inflammatory cytokine secretion, further fueling the inflammatory cascade. Other pro-inflammatory prostanoids like PGE2 and PGI2 are predominant in contributing to increased vascular permeability and blood flow in the inflamed region, leading to fluid leakage. TXA2, for its part, can increase platelet-leukocyte interactions, forming microvascular clots and contributing to the thrombotic complications seen in COVID-19. Interestingly, PGD2, through specific receptors, may paradoxically contribute to the resolution of inflammation, showing the dual or opposing nature of these molecules even within the inflammatory response itself.
What about lymphocytes, the central players in adaptive immunity? Do eicosanoids affect them directly, and how does this influence the broader immune response?
Lymphocytes, while central to adaptive immunity, have minimal capacity to form prostaglandins themselves, yet they are a primary target of prostaglandin action, making them highly susceptible to external eicosanoid signals. Prostaglandins generally inhibit lymphocyte function and proliferation, thereby suppressing aspects of the adaptive immune response. PGE2 plays a particularly complex and nuanced role. It depresses the humoral antibody response by inhibiting the differentiation of B lymphocytes into antibody-secreting plasma cells. At the same time, it can induce immunoglobulin class switching, specifically resulting in increased IgE production, which is relevant in allergic responses. PGE2 also acts on T lymphocytes to inhibit mitogen-stimulated proliferation and lymphokine release by sensitized cells, effectively dampening their activation and function. Furthermore, PGE2 and TXA2 may also play a critical role in T lymphocyte development by regulating apoptosis or programmed cell death of immature thymocytes, shaping the T-cell repertoire.
And there's a truly cutting-edge connection here to cancer immunotherapy, specifically with PD-1 and PD-L1 pathways. Can you elaborate on that? This sounds huge.
This is an absolutely crucial and highly active area of research. PGE2, acting via its EPR2 and EPR4 receptors, can interact with programmed cell death protein 1, or PD-1, a key inhibitory checkpoint on immune cells, to restrain cytotoxic T-cell function and survival during chronic infection, which mirrors the chronic inflammatory state of a tumor. Furthermore, the COX2 microsomal PGE synthase 1 PGE2 pathway can directly regulate the expression of PD ligand 1, or PDL1, on tumor-infiltrating myeloid cells. Given the established and transformative efficacy of blocking the PD-1, PDL1 pathway in a wide range of cancers, a cornerstone of modern immunotherapy, this strongly suggests that blockade of PGE2 synthesis or its action might actually augment the efficacy of PD-1, PDL1 blockade in cancer treatment. This is a very promising therapeutic strategy currently under intense investigation.
That's a profound connection to a major cancer treatment strategy, offering potential synergistic therapies. Beyond that, what specific roles do eicosanoids play in leukocyte chemoattraction and activation, literally directing the body's immune cells to where they need to go?
Well, PGD2, through its DPR2 receptor, is a potent leukocyte chemoattractant, meaning it actively draws immune cells to specific locations, a critical first step in inflammation. Leukotrienes are also incredibly potent mediators of inflammation. Genetic deletion of either 5-LOX or its activating protein, FLAP, significantly reduces inflammatory responses in various model systems, unequivocally underscoring their importance in orchestrating these responses. LTB4, in particular, is a profoundly pro-inflammatory eicosanoid. It acts as a powerful chemotactic agent for neutrophils, T lymphocytes, eosinophils, monocytes, dendritic cells, and possibly mast cells, actively recruiting them to sites of injury or inflammation. LTB4 also stimulates the aggregation of eosinophils and promotes their degranulation, releasing inflammatory mediators and the generation of superoxide, a reactive oxygen species. It further promotes the adhesion of neutrophils to vascular endothelial cells and their transendothelial migration, which is essential for immune cells to leave the bloodstream and enter inflamed tissue. Moreover, LTB4 stimulates the synthesis of co-inflammatory cytokines from macrophages and lymphocytes, amplifying the inflammatory cascade.
And the cystinyl leukotrienes, LTC4, LTD4, LTE4, what are their specific contributions to leukocyte activation and the inflammatory response?
Cyclooxygenases are also potent chemotaxins for eosinophils and monocytes, drawing these specific cell types to sites of inflammation. They also induce cytokine generation in eosinophils, mast cells, and dendritic cells, contributing to the inflammatory signaling network. At higher concentrations, cyclooxygenases promote eosinophil adherence, degranulation, cytokine or chemokine release, and oxygen radical formation, all processes that contribute to tissue damage and inflammation. In addition, cyclooxygenases further contribute to inflammation by increasing endothelial permeability, a process that allows fluid and inflammatory cells to leak out of blood vessels and into the inflamed tissue, thereby promoting the migration of even more inflammatory cells to the site of inflammation. This intricate network of actions shows how eicosanoids finally control the movement and activation of virtually all major immune cells.
Let's shift our focus to the bronchial and tracheal muscle. How do eicosanoids profoundly influence our breathing, and how does this relate to conditions like asthma?
When sensitized lung tissue is challenged by an appropriate antigen, such as an allergen, a complex mixture of autacoids is rapidly released, including both COX-derived bronchodilator and bronchoconstrictor substances. Specifically, TXA2, PGF2, and PGD2 are potent bronchoconstrictors, causing the smooth muscle surrounding the airways to contract and leading to narrowing of the air passages. On the other hand, PGE2 and PGA2 are powerful bronchodilators, relaxing bronchial and tracheal muscle, thereby opening up the airways. PGI2 in particular causes profound bronchodilation in most species. Human bronchial tissue is especially sensitive to its effects. PGI2 also actively antagonizes bronchoconstriction induced by other agents and suppresses allergic airway inflammation by promoting regulatory T-cell signaling and reprogramming, acting as a crucial anti-inflammatory agent in the lungs. PGD2 appears to be the primary bronchoconstrictor prostanoid of clinical relevance in humans, especially in allergic reactions.
And this ties directly into conditions like asthma, right? Especially that specific and challenging condition known as aspirin-exacerbated respiratory disease.
Absolutely. The genetic predisposition to asthma can even be linked to eicosanoid pathways. Polymorphisms or genetic variations in the genes for hematopoietic PGE2 synthase, or HPGDS, and the TPR have been associated with asthma in humans. And a very specific condition affecting roughly 10% of people who take aspirin or NSAIDs is the development of bronchospasm, a condition specifically termed aspirin-exacerbated respiratory disease, or AERT. This immune dysfunction involves a profound shift in arachidonic acid metabolism. Because COX-1 is inhibited, the substrate is diverted to the LOX pathway, leading to an overproduction of leukotrienes. This substrate diversion appears to primarily involve COX-1, not COX-2, making non-selective NSAIDs particularly problematic for these patients. Cysteine leukotrienes are incredibly potent bronchoconstrictors, 1,000 times more potent than histamine acting principally on smooth muscle in the airways. They also stimulate bronchial mucus secretion and cause mucosal edema, all contributing to the severe symptoms of asthma.
So cyclooxygenases are indeed key players in allergic constriction of the airway.
They profoundly dominate during allergic constriction of the airway. This is further supported by the fact that deficiency of 5-LOX, the enzyme that starts leukotriene synthesis, leads to reduced eosinophil influx in airways and significantly attenuates bronchoconstriction in models. Furthermore, unlike traditional COX inhibitors and histaminergic antagonists, cysteine-TR antagonists and 5-LOX inhibitors are highly effective in the treatment of human asthma, highlighting the critical role of leukotrienes in this disease. The relatively slow leukotriene metabolism in the lung contributes significantly to the long-lasting bronchoconstriction that often follows antigen challenge and may be a factor in the chronically high bronchial tone observed in asthmatic patients, even between acute attacks.
Shifting to the gut, how do eicosanoids regulate GI smooth muscle and overall gut motility? It sounds like they have a strong influence on digestion.
In the gastrointestinal tract, eicosanoids play a nuanced but potent role in regulating smooth muscle function. PGE2 and PGF2s stimulate the contraction of the main longitudinal muscle layer throughout the GI tract, from the stomach to the colon. Prostaglandin and doperoxides, TXA2 and PGI2, also produce contraction but are generally less active than PGE2 and PGF2. Interestingly, the circular muscle layer, which runs perpendicular to the longitudinal muscle, generally relaxes in response to PGE2 and contracts in response to PGF2, showcasing differential effects even within the same organ and highlighting the intricate controlled gut movements. The leukotrienes also have potent contractile effects on GI smooth muscle. Overall, prostaglandins significantly reduce transit time in the small intestine and colon, essentially speeding up the movement of contents. This can manifest clinically. Diarrhea, abdominal cramps, and reflux of bile have all been noted in response to oral PGE analogs. Furthermore, PGE2 and PGF2 stimulate the movement of water and electrolytes into the intestinal lumen, which is why watery diarrhea can follow their oral or parenteral administration. PGE2 appears to contribute significantly to the water and electrolyte loss observed in cholera, a severe diarrheal disease, which is why it's somewhat responsive to therapy with NSAIDs that block PGE2 synthesis, further highlighting the role of eicosanoids in fluid balance.
And what about GI secretion, particularly the stomach's critical cytoprotective shield against its own harsh acid?
In the stomach, PGE2 and PGI2 are absolutely crucial for contributing to increased mucus secretion, which forms a thick protective layer on the stomach lining, a key aspect of what we call cytoprotection. They also contribute to reduced acid secretion and reduced pepsin content, both of which lessen the corrosive impact on the gastric mucosa. PGE2 and its analogues also powerfully inhibit gastric damage caused by various ulcerogenic agents, such as NSAIDs themselves, and actively promote the healing of both duodenal and gastric ulcers. While COX-1 may be the dominant source of such cytoprotective PGs under normal physiological conditions, both COX-1 and COX-2 may contribute to ulcer healing through the regulation of angiogenesis or the formation of new blood vessels. PDE2 specifically promotes the healing of GI ulcers through the stimulation of angiogenesis via the upregulation of vascular endothelial growth factor expression, which is mediated by its EPR4 receptor, literally helping to rebuild damaged tissue.
So inhibiting COX-2, as with the COXs, could actually impair ulcer healing, which would be a surprising unintended consequence.
That is indeed a significant concern, although its clinical impact in humans is still being clarified. Selective inhibitors of COX-2 and genetic deletion of the enzyme delay ulcer healing in rodents, suggesting a potential impairment. Conversely, LTs, by constricting gastric blood vessels and enhancing the production of pro-inflammatory cytokines, may actively contribute to gastric damage, showcasing yet another layer of opposing forces and balances in the gut.
Moving to the uterus, it's clear eicosanoids play a vital and incredibly dynamic role there, from regulating the menstrual cycle to orchestrating the profound events of childbirth.
Indeed. The uterus is a remarkable example of eicosanoid control. Strips of non-pregnant human uterus are potently contracted by PGF2 and TXCE2, but are relaxed by PGE2, showcasing opposing actions. The sensitivity of the uterus to the contractile response is most prominent before menstruation, whereas relaxation is greatest at mid-cycle, indicating how closely their actions are tied to hormonal fluctuations throughout the menstrual cycle. Critically, PGE2, together with oxytocin, is absolutely essential for the onset of parturition or childbirth. Conversely, PGI2 and high concentrations of PGE2 actually produce uterine relaxation, highlighting a nuanced balance during pregnancy. The intravenous infusion of even low concentrations of PGE2 or PGF2 to pregnant women produces a dose-dependent increase in uterine tone and in the frequency and intensity of rhythmic uterine contractions. This profound effect is therapeutically harnessed, as PGE2 and PGF2 can be administered to induce labor at term or to terminate pregnancy. Uterine responsiveness to prostaglandins generally increases as pregnancy progresses, but remains smaller than the more powerful response to oxytocin. The critical role of PGF2 in parturition specifically relies on its capacity to induce an oxytocin-dependent decline in progesterone levels, which is a key hormonal shift that facilitates the onset and progression of labor.
What about the kidney? How do eicosanoids regulate crucial fluid balance and blood pressure in such a fundamental organ?
Both COX-1 and COX-2 derived eicosanoids are absolutely crucial regulators of kidney blood flow and the glomerular filtration rate, or GFR, which is how well your kidneys are filtering waste. COX-2 drive PGE2 and PGI2 increase medullary blood flow, leading to what's called pressure diuresis, increased urine output due to increased blood flow, and they also inhibit tubular sodium reabsorption, promoting salt excretion. The expression of medullary COX-2 is even increased during high salt intake, demonstrating its adaptive role in salt regulation. COX-1-derived products, on the other hand, promote salt excretion in the collecting ducts, showing a complementary role. In the renal cortex, COX-2-derived PGE2 and PGI2 increase renal blood flow and GFR through their local vasodilating effects and as part of the tubuloglomerular feedback mechanism that precisely controls renin release. This is important because expression of COX-2 in macula densa cells increases in conditions of low distal tubular flow, such as during low dietary salt intake or volume depletion. COX-2-derived PGE2 and possibly PGI2 then result in increased renin release, which ultimately leads to sodium retention and can contribute to elevated blood pressure, demonstrating their tight connection to the renin-angiotensin system.
And do TXA2 or PGF2 also play a role in kidney function, perhaps with opposing effects?
Yes, they do. TXA2, although generated at low levels in the normal kidney, has potent vasoconstrictor effects that can reduce renal blood flow and GFR. Infusion of PGF2 causes both natriuresis, or sodium excretion, and diuresis, water excretion. Conversely, PGF2 may also activate the renin-angiotensin system, potentially contributing to elevated blood pressure. Furthermore, the CYP epoxygenase products, the ETs we discussed, also play a role in regulating renal function. Both 20-HEAT and the ETs are generated in renal tissue. 20-HEAT constricts the renal arteries, while ETs mediate vasodilation and natriuresis, again highlighting opposing local effects.
And there's a specific rare syndrome linked to this intricate renal regulation, right? Bartter syndrome.
That's right. Bartter syndrome is an autosomal recessive genetic trait manifesting as hypokalemic metabolic alkalosis, a complex electrolyte imbalance. The antenatal variant, which manifests before birth, is due to dysfunctional RMK2, a specific potassium channel in the kidney. This syndrome is notably also known as hyper-PGE syndrome because patients exhibit abnormally elevated PGE2 synthesis. While the precise relationship between dysfunctional RMK2 and elevated PGE2 synthesis isn't fully clear, the clinical connection is striking. Inhibition of COX2 in patients with antenatal Bartter syndrome ameliorates many of the severe clinical symptoms, highlighting a critical therapeutic connection and underscoring the profound impact of PGE2 on renal function and systemic electrolyte balance.
Moving to the eye, how do eicosanoids influence intraocular pressure, and are they therapeutically relevant for conditions like glaucoma, a leading cause of blindness?
PGF2 induces constriction of the iris sphincter muscle, but its overall and crucially important effect in the eye is actually to decrease intraocular pressure by increasing aqueous humor outflow, the drainage of fluid from inside the eye. This effect is very therapeutically relevant, as a variety of FPR agonists, which mimic the action of PGF2, have proven highly effective in the treatment of open-angle glaucoma, a condition often associated with the loss of COX2 expression in the pigmented epithelium of the ciliary body. This suggests a potential link between reduced COX2 and increased intraocular pressure. Furthermore, a newer EPR2 agonist, which has a non-prostaglandin structure but activates the PGE2 receptor, has recently been approved for the treatment of glaucoma and ocular hypertension, demonstrating ongoing innovation and diversified approaches in this area.
What about the central nervous system? Do eicosanoids affect everything from inducing fever to influencing cognition and sleep? That's an astonishing range.
Absolutely. The CNS is profoundly influenced by eicosanoids. PGE2 is the key mediator well known for inducing fever. The hypothalamus regulates the body temperature set point, and this set point is elevated by endogenous pyrogens, fever-inducing substances like interleukin, IL-1, IL-6, TNF-α, and interferons, which are released during infection or inflammation. The fever response is mediated by the coordinated induction of COX2 and microsomal PGE synthase 1, or MPGES1, in the endothelium of blood vessels in the preoptic hypothalamic area. This leads to a rapid local formation of PGE2. PGE2 can then cross the blood-brain barrier and act on EPR3, and perhaps EPR1, on thermosensitive neurons, triggering the hypothalamus to elevate body temperature. While exogenous PGF2 and PGI2 can induce fever, they don't appear to contribute to the body's endogenous pyretic response, highlighting PGE2's specific role.
So PGE2 is the master fever inducer. What about its less obvious roles, like in cognitive aging? That sounds really interesting.
Right. PGE2 can play a maladaptive role in age-associated inflammation and cognitive decline. It does this through its myeloid EPR2 receptor, which can induce an energy-deficient state in macrophages and microglia, the brain's immune cells. Crucially, EPR2 blockade has been shown to reduce this inflammatory response, restore cellular bioenergetics, and even reverse cognitive aging in preclinical models, opening up exciting avenues for treating age-related cognitive impairment. As for sleep, PGD2 appears to act on arachnoid trabecular cells in the basal forebrain to mediate an increase in extracellular adenosine that, in turn, facilitates the induction of sleep, suggesting a role in normal sleep regulation. COX2-derived prostanoids have also been implicated in the pathogenesis of several central nervous system degenerative disorders, such as Alzheimer's disease and Parkinson's disease, where inflammation is increasingly recognized as a contributing factor.
And pain. This is a massive area, isn't it, given their role in inflammation?
Indeed. Eicosanoids play a critical amplifying role in pain perception. Inflammatory mediators, including leukotrienes and prostaglandins, significantly increase the sensitivity of nociceptors, which are your pain receptors, and thereby potentiate pain perception. Centrally, both COX1 and COX2 are expressed in the spinal cord under basal conditions and release prostaglandins in response to peripheral pain stimuli, indicating their role in spinal pain processing. Peripherally, both PGE2 through EPR1 and EPR4 and PGA2 via IPR reduce the threshold to stimulation of nociceptors, causing what we call peripheral sensitization. This means they make the nerve endings that detect pain more sensitive to even mild stimuli.
So they don't necessarily cause the initial pain, but they make you feel it much more acutely, like turning up the volume on the pain signal?
Precisely. That's a good way to put it. PGE2 and perhaps PGD2, PGI2, and PGF2A can increase excitability in neuronal pain transmission pathways in the spinal cord, leading to hyperalgesia, which is an increased sensitivity to painful stimuli, and allodynia, where normally non-painful stimuli are perceived as painful. LTB4 also directly produces hyperalgesia. The release of these eicosanoids during the inflammatory process thus serves as a powerful amplification system for the pain mechanism, making inflammatory pain a significantly more intense and pervasive experience. Their role in inflammatory pain is so critical that it's often a primary target for pain relief therapies.
What about the endocrine system? How do eicosanoids influence hormone release and glandular function?
Well, systemic administration of PGE2 significantly increases circulating concentrations of several key hormones, including ACTH, corticotropin, growth hormone, prolactin, and gonadotropins, showcasing a broad influence on the master endocrine glands. Other direct effects include the stimulation of steroid production by the adrenals, stimulation of insulin release from the pancreas, and even thyrotropin-like effects on the thyroid gland. In reproduction, PGE2 works as part of a positive feedback loop, essential to induce oocyte maturation required for fertilization during and after ovulation. This process is finally regulated by the activity of the prostaglandin transporter, PGT, which precisely modulates extracellular PGE2 levels in granulosa cells, ensuring the right amount of signal at the right time. We also discussed how the critical role of PGF2 in parturition relies on its capacity to induce an oxytocin-dependent decline in progesterone levels, a crucial hormonal shift for labor. Even LOX metabolites have endocrine effects. 12-HEAT, for example, stimulates the release of aldosterone from the adrenal cortex and mediates a portion of the aldosterone release stimulated by angiotensin II, but notably not that which occurs in response to ACTH.
They're involved in bone metabolism too, right? Both building and breaking bone. That sounds like a constant balancing act.
Yes, prostaglandins are powerful modulators of bone metabolism. COX1 is expressed constitutively in normal bone, playing a role in its baseline maintenance, while COX2 is significantly upregulated in settings such as inflammation and during mechanical stress, indicating its dynamic involvement in bone remodeling and repair. PGE2 uniquely stimulates both bone formation by increasing osteoblastogenesis, the creation of new bone cells, and bone resorption via activation of osteoclasts, which break down bone. This dual action is absolutely crucial for continuous bone remodeling and maintaining bone homeostasis. Furthermore, recent research shows that osteoblast-derived PGE2 activates EPR4 receptors in sensory nerves to control bone homeostasis and regeneration, highlighting a neurobone axis.
And skeletal muscle, from repair after injury to overall regeneration and maintenance.
Prostaglandins play an essential and dynamic role in skeletal muscle biology. PDE2 is the main prostaglandin produced in resting skeletal muscle, suggesting a basal regulatory function. It contributes significantly to the regeneration of damaged muscles and maintains the homeostasis of mature muscle myofibers. Acting via its EPR4 receptor, PGE2 powerfully drives muscle repair after injury by accelerating muscle stem cell expansion, which is critical for rebuilding muscle tissue. Consequently, COX2 deletion or inhibition may impair skeletal muscle healing, which is an important consideration for athletes or those recovering from injury. In aging skeletal muscle, the inactivation of 15-PGDH, an enzyme whose expression increases with advancing age, has been shown to restore PGE2 and PGD2 levels, leading to increased muscle mass, strength, and exercise performance by positively impacting mitochondrial function, autophagy, and proteostasis, offering potential strategies for combating age-related muscle decline or sarcopenia. HPGDS-derived PGD2 is also implicated in muscle necrosis and muscle tissue damage. In fact, HPGDS inhibition ameliorates myonecrosis in mouse models of Duchenne muscular dystrophy, and urinary excretion of PGD2 metabolites reflects the severity of this disease, making it a potential biomarker.
Finally, a very impactful and complex area, cancer. How are eicosanoids involved in pro-oncogenic roles, and what are the profound implications for modern immunotherapy?
This is a truly significant and active area of research. Pharmacological inhibition, or genetic deletion of COX2, has been consistently shown to restrain tumor formation in models of colon, breast, lung, and other cancers, indicating its crucial role in tumorigenesis. Large human epidemiological studies have reported that incidental use of NSAIDs, which, remember, inhibit COX, is associated with significant reductions in the relative risk for developing these and other cancers, strongly suggesting a protective effect. PGE2 has been broadly implicated as the primary pro-oncogenic prostanoid in multiple studies. PGE2, whether it's produced by the tumor cell itself, by stromal cells within the tumor microenvironment, or by infiltrating immune cells, can actively promote cancer initiation and progression by controlling multiple fundamental biological processes.
So it's not just the tumor cells themselves, but also their surrounding environment that contributes to this pro-cancer effect. It's the whole neighborhood.
Exactly. That's a great way to think about it. For instance, fibroblast-derived PGE2 may induce early tumor initiation by driving the proliferation of epithelial stem cells through EPR4. Deletion or inhibition of EPR4 significantly reduces tumor formation in mouse models of colon cancer, highlighting a specific pathway. PGE2 derived from COX2 can further promote tumor progression by actively shaping the tumor microenvironment and, crucially, by favoring tumor escape from immune surveillance. This is where the profound connection to immunotherapy becomes truly fascinating. COX2 deletion demonstrably reduces tumor growth by restoring effector CD8 plus T-cell infiltration. These are the killer T-cells that fight cancer, and by reducing myeloid-derived suppressor cells, which are immune cells that normally dampen anti-tumor immunity. COX2 inhibition has even been shown to reverse tumor resistance to immunotherapy in mouse models of pancreatic adenocarcinoma, a notoriously difficult-to-treat cancer.
That's a direct link to improving current cancer treatments. How specifically does PGE2 affect immune evasion, essentially helping cancer hide from our immune system?
Well, PGE2 induces cytotoxic T lymphocyte exhaustion by modulating inhibitory T-cell receptor function, making these crucial anti-cancer cells less effective and tired. It can directly upregulate PD-1 expression on effector CD8 plus T-cells and PD-L1 expression in tumor-associated macrophages and myeloid-derived suppressor cells. Remember, PD-1 and PD-L1 are the off switches that cancer cells use to evade immune attack. PGE2 also controls the suppressive activity of polymorphonuclear myeloid-derived suppressor cells, PMN-MDSCs, which dampen the anti-tumoral activity of T cells and other immune cells. Furthermore, PGE2 reduces the viability of natural killer cells, their chemokine production, and chemokine receptor expression in conventional type 1 dendritic cells, all key players in initiating an effective anti-tumor immune response. Given the established and transformative efficacy of blockade of the PD-1-PD-L1 pathway in a range of cancers, the clear possibility that blockade of PGE2 synthesis or action might augment this effect has been strongly suggested and is a major focus for future therapeutic strategies.
Okay, let's pivot now. Section 6, Platelet-Activating Factor, PAF. Now that we've taken a comprehensive look at the intricate world of eicosanoids, let's shift our focus to another equally critical lipid-derived autacoid, platelet-activating factor, or PAF. Its name suggests a primary role with platelets, but I understand its actions extend far, far beyond that.
You're absolutely right. While its name focuses on platelets, PAS is a profound pleiotropic mediator, meaning it exerts multiple diverse effects across a wide range of cell types and physiological systems. Its intriguing discovery journey began in 1971 when Robert Henson, an American immunologist, demonstrated that a soluble factor released from leukocytes' white blood cells caused platelets to aggregate. This was an exciting initial observation that pointed to a new type of biological messenger. Then, Jacques Benveniste and his coworkers, building on this, characterized this factor as a polar lipid and gave the descriptive name PAF.
So it was identified by its function, its ability to activate platelets, before its precise chemical structure was fully known?
Yes, that's exactly right. And around the same time, remarkably, there were parallel discoveries that would confirm its identity. Eric Muirhead, an American nephrologist, for instance, described an antihypertensive polar renal lipid, or APRL, produced by renal medullary interstitial cells, which was subsequently proven to be chemically and biologically identical to PAF. Concurrently, Donald Hanahan and his co-workers in the U.S. synthesized acetylglycerol ether phosphorylcholine, or AGPC, and found that this synthetic phospholipid had identical chemical and biological properties to PAF. So despite these multiple independent discoveries and different names, the commonly accepted name for this substance is PAF, even though, as you noted, its actions extend far beyond just activating platelets. Its broad impact makes it a truly fascinating molecule.
Let's delve into the chemistry and biosynthesis of PAF. What's its unique structure, and how is it made so precisely by the body?
PAF, chemically known as 1-O-alkyl-2-acetyl-ethylglycerol-3-phosphocholine, actually represents a family of phospholipids because the alkyl group at position 1 can vary in length, typically from 12 to 18 carbon atoms. The structural variability can influence its specific biological activity. In human neutrophils, for example, PAF predominantly consists of a mixture of the 16 and 18 carbon ethers, but its precise composition can change upon cell stimulation, adapting to different physiological conditions. Similar to eicosanoids, the biosynthesis of PAF also profoundly depends on PLA2 activity for the crucial initial step of precursor release.
And what's the major biosynthetic pathway for PAF? How does the body control its production?
The primary biosynthetic pathway for PAF is called the remodeling pathway. It involves a specific membrane lipid precursor, 1,0-alkyl-2-acyl-glycerophosphocoin, where the 2-acyl substituents can include arachidonic acid, linking it to the eicosanoid system. PAF is synthesized from this precursor in two key enzymatic steps. First, PLA2 removes the acyl group at position 2, forming an intermediate compound called lyso-PAF. Then in the second and crucial step, an enzyme called acetyl coenzyme acyl PAF, acetyltransferase, adds an acetyl group, forming the active PAF molecule. This second step, catalyzed by acetyltransferase, is the rate-limiting step for PAF biosynthesis. This means it controls the overall speed and amount of PAF produced, making it a critical point for physiological regulation.
So that second step is where the body controls how much PAF is produced, ensuring it's made only when needed. What kinds of stimuli trigger the synthesis?
PAF synthesis can be stimulated during antigen-antibody reactions, which are typical allergic or immune responses, or by various other potent agents, including chemotactic peptides, thrombin, collagen, and even other autacoids, highlighting its role in diverse physiological and pathological processes. Interestingly, PAF can also stimulate its own formation, creating a positive feedback loop under certain conditions that can amplify its effects. Both the phospholipase and acetyltransferase enzymes involved in this pathway are calcium-dependent enzymes, meaning that PAF synthesis is tightly regulated by the availability of intracellular calcium, adding another layer of control.
And how is PAF broken down once it's done its job, similar to eicosanoids?
PAF is efficiently and rapidly inactivated by a family of enzymes called PAF acetylhydrolases, or PAF-AHs. These enzymes catalyze the precise hydrolysis of the acetyl group at the SN2 position, generating lyso-PAF, which is then converted back to a 1,0-alkyl-2-acylglycerophosphocholane by an acetyltransferase. This efficient degradation ensures that PAF's effects are transient and localized, preventing prolonged or excessive signaling. Beyond this remodeling pathway, PAF synthesis can also occur de novo, or from scratch, by transferring a phosphocholine substituent to alkyl acetylglycerol via a lysoglycerophosphate acetyl coenzyme A transferase. This pathway may contribute to the low basal physiological levels of PAF needed for normal cellular functions separate from acute responses.
Are there other PAF-like molecules that mimic its actions, especially those related to oxidative stress?
Yes, this is an important point. PAF-like molecules can indeed be formed from oxidized phospholipids, or OXPLs. These compounds are significantly increased in settings of oxidant stress, such as from cigarette smoking, highlighting a link between environmental factors and lipid mediators. They differ structurally from PAF in that they contain a fatty acid at the SN1 position of glycerol joined through an ester bond, and various short-chain acyl groups at the SN2 position. However, despite these structural differences, OXPLs effectively mimic PAF structure, bind to its receptor, and crucially, elicit the same biological responses, making them functional mimics. The key distinction is that, unlike the highly controlled enzyme-driven synthesis of PAF, OXTL production is largely unregulated, which can contribute to pathological conditions where oxidative stress is prominent. Degradation of OXPLs by PAF-AH is necessary to suppress their toxicity, and interestingly, increased levels of plasma PAF-AH have been reported in conditions like colon cancer, cardiovascular disease, and stroke, suggesting a compensatory response to high OXPLs or perhaps a role in the pathology itself.
So, if PAF isn't stored, where is it made, and how does it act locally to avoid widespread effects?
Since PAF is not stored in cells, it is synthesized rapidly and precisely in response to stimulation, acting as that immediate on-demand messenger. It's produced by a wide range of cell types, including platelets, neutrophils, monocytes, mast cells, eosinophils, renal mesangial cells, renal medullary cells, and vascular endothelial cells, all cells intimately involved in inflammation or vascular regulation. Depending on the cell type and the specific stimulus, PAF can either remain tightly cell-associated or be secreted. For example, it's readily released from monocytes, but it's often retained by leukocytes and endothelial cells. In endothelial cells, for instance, PAF is not freely secreted, but is displayed on the cell surface for what we call juxtacrine signaling. This means it stimulates adherent leukocytes directly without being released into general circulation. This pro-adhesive function of PAF is incredibly important for orchestrating the initial interaction of platelets and circulating inflammatory cells with inflamed endothelium, often acting cooperatively with P-selectin, enabling immune cells to roll and stick to the vessel wall at sites of injury.
That makes a lot of sense for localized inflammation, ensuring the response is precise. How does extracellular PAF transmit its signals to recipient cells? What's its detailed mechanism of action?
Extracellular PAF exerts its potent actions by stimulating a specific G protein-coupled receptor, the PAF receptor. This receptor is a classic GPCR and couples to two main intracellular G proteins. GQ, which activates the phospholipolysis inositol 1004-5-trisphosphate calcium pathway, leading to a rapid increase in intracellular calcium, and G, which inhibits adenyl cyclase, thereby reducing cyclic AMP levels. The consequent activation of other phospholipases like PLA2, PLC, and PLD gives rise to additional second messengers, including AA-derived prostaglandins, TXA2, or leukotrienes, which may further mediate and amplify the effects of PAF, creating a complex and interconnected signaling network.
Does it also activate other critical cellular pathways, like those involved in cell growth and gene expression?
Yes, further downstream, P38 mitogen-activated protein kinase is activated following the PAF receptor G-key interaction, a key step in stress and inflammatory signaling. Extracellular signal-regulated kinase, or ERK, activation can also occur. This can happen via direct interaction of the activated PAF receptor with GQ, GO, or their beta-gamma subunits, or even via transactivation of the epidermal growth factor receptor, ultimately leading to nuclear factor key, NF key activation, a master regulator of inflammation and gene expression. The crystal structure of the PAF receptor was recently resolved, providing critical structural insights for rational drug design and potentially more targeted therapies. Crucially, as we touched on earlier, PAF exerts many of its important pro-inflammatory actions without ever leaving its cell of origin. For instance, PAF synthesized in a regulated fashion by endothelial cells, stimulated by inflammatory mediators, is presented on the endothelium surface, where it directly activates the PAF receptor on juxtaposed cells like platelets, PMNs, and monocytes, fostering that crucial adhesion that's foundational for the inflammatory response.
So PAF truly is a pleiotropic mediator, affecting so many different systems and processes in the body. What are its main physiological and pathological functions, revealing its broad impact on health and disease?
PAF is indeed a remarkably potent lipid mediator of pathological events and has been implicated in a wide range of significant conditions. These include allergic asthma, endotoxic shock, a severe life-threatening response to bacterial toxins, acute pancreatitis, certain cancers, dermal inflammation, and chronic inflammatory cardiovascular diseases like atherosclerosis.
Given its potent effects, especially in inflammation and allergy, have PAF antagonist drugs that block its action been successful in treating inflammatory or allergic disorders in humans? That seems like a logical step.
While experimental administration of PAF can impressively reproduce many of the acute signs and symptoms observed in anaphylactic shock, a severe allergic reaction, the clinical translation for PAF antagonists has unfortunately been disappointing. Despite extensive research, none has proven broadly clinically useful to date for these conditions. For example, in asthma patients, PAF antagonists only partially inhibit the bronchoconstriction induced by antigen challenge, meaning they don't fully block allergic asthma attacks. Crucially, they have no effect on bronchoconstriction induced by challenges from methacholine, exercise, or cold air. This lack of broad efficacy likely reflects the inherent complexity of these pathological conditions and the high probability that many other mediators also contribute significantly to the inflammation and symptoms associated with these disorders. It's rarely just one molecule,
Okay, fair enough. Let's move to the cardiovascular system. What are PAF's specific effects there, both on blood vessels and heart function?
PAF is a potent vasodilator in most vascular beds. When administered intravenously, even at low doses, it causes hypotension, a drop in blood pressure. This vasodilation is notably independent of sympathetic innervation, the renin-angiotensin system, or arachidonic acid metabolism, suggesting it acts through unique direct and indirect actions. However, PAF can also induce vasoconstriction, depending on the concentration, the specific vascular bed, and importantly, the involvement of platelets or leukocytes. For example, very low intracoronary PAF increases coronary blood flow via platelet-derived vasodilator, but higher doses can paradoxically decrease it, likely through the formation of intravascular platelet aggregates or TXA2 formation, leading to blockages. The pulmonary vasculature is also constricted by PAF through a similar mechanism. Intradermal injection of PAF causes an initial transient vasoconstriction followed by a typical wheal and flare response on the skin, characteristic of allergic reactions. Most dramatically, PAF profoundly increases vascular permeability and edema by causing contraction of venular endothelial cells. In this respect, its actions resemble those of histamine and bradykinin, but PAF is orders of magnitude more potent, about 1,000-fold more potent than histamine, making it a major player in fluid leakage during inflammation.
And with platelets, it's in its name, platelet-activating factor. How does it affect them, and how does this relate to clotting?
Right. The PAF receptor is constitutively expressed on the surface of platelets, meaning it's always present and ready to respond. PAF potently stimulates platelet aggregation. When injected intravenously, PAF causes the rapid formation of intravascular platelet aggregates and thrombocytopenia, a reduction in platelet count by clumping them together. While this aggregation is accompanied by the release of TXA2 and granular contents, PAF does not require the presence of TXA2 or other aggregating agents to produce its effect, highlighting its independent and powerful aggregating ability. However, it's important to note that PAF antagonists fail to block thrombin-induced aggregation, even though they can prolong bleeding time and prevent thrombus formation in some experimental models. This indicates that PAF contributes significantly to thrombus formation by recruiting and activating platelets, but it does not function as the sole independent primary mediator of all platelet aggregation. It's a powerful accessory, but not always the initiator.
What about its critical role in leukocytes, the inflammatory cells? How does PAF activate and recruit them to sites of inflammation?
Platelet-activating factor is a potent and very common activator of inflammatory cells, stimulating a wide variety of responses in polymorphonuclear leukocytes, PMNs, which include eosinophils, neutrophils, and basophils. PAF stimulates PMNs to aggregate, degranulate, releasing their stored inflammatory mediators, and generate free radicals and leukotrienes, all contributing to the inflammatory cascade. It is also a potent chemotactic factor for eosinophils, neutrophils, and monocytes, actively recruiting these crucial immune cells to sites of inflammation. It powerfully promotes PMN endothelial adhesion, contributing, along with other adhesion molecular systems, to leukocyte rolling, tight adhesion, and then their essential migration through the endothelial monolayer to reach the inflamed tissue. PAF also stimulates basophils to release histamine, activates mast cells, and induces cytokine release from monocytes. In addition, it promotes the aggregation of monocytes and the degranulation of eosinophils. It truly is central to mobilizing and orchestrating the entire immune response.
So it's a master orchestrator of inflammation. What about its effects on smooth muscle throughout the body, beyond just blood vessels?
PAF potently contracts various types of smooth muscle, including those in the gastrointestinal tract, the uterus, and the pulmonary airways. In the uterus, it significantly enhances the amplitude of spontaneous uterine contractions, which notably are inhibited by inhibitors of prostaglandin synthesis, suggesting an interaction between these two lipid mediator systems. While PAF does not affect tracheal smooth muscle, it robustly contracts airway smooth muscle, leading to narrowing. When given by aerosol, PAF increases airway resistance as well as responsiveness to other bronchoconstrictors, making it a major player in respiratory conditions like asthma. PAF also increases mucus secretion and the permeability of pulmonary microvessels, further contributing to the symptoms seen in lung inflammation.
And its effects on the stomach. You mentioned earlier that it's an incredibly potent ulcerogen.
Yes. In addition to contracting the fundus of the stomach, PAF is actually the most potent known ulcerogen discovered to date. When given intravenously, even at very low doses, it causes hemorrhagic erosions of the gastric mucosa that extend deeply into the submucosa, highlighting its severe potential for inducing gastric damage and bleeding. This makes it a significant molecule in understanding gastrointestinal pathology.
What about its impact on the kidney, beyond just blood flow?
PAF profoundly affects renal function. It decreases renal blood flow, glomerular filtration rate, urine volume, and the excretion of sodium, and it does so often without changes in systemic hemodynamics, suggesting a direct renal effect. It exerts a receptor-mediated biphasic effect on afferent arterioles, the tiny blood vessels leading into the kidney's filtering units, dilating them at very low concentrations and constricting them at higher concentrations. The vasoconstrictor effect appears to be mediated, at least in part, by COX products, whereas vasodilation is a consequence of stimulated nitric oxide production by endothelium. This complex biphasic action fine-tunes renal blood flow.
Any other significant functions or implications for PAF, especially in broader diseases like cancer?
Yes, certainly. PAF is a potent mediator of angiogenesis, the formation of new blood vessels, which is a critical process in wound healing, but also, unfortunately, in tumor growth and metastasis. For this reason, PAF has been implicated in the progression of many cancers, including breast, prostate, esophageal, lung, pancreatic, and skin cancers. Interestingly, cigarette smoke exposure has been found to upregulate PAF signaling, potentially linking this common environmental factor to various diseases via PAF. Conversely, PAF-AH deficiency, meaning a genetic lack of the enzyme that breaks down PAF, has been associated with small increases in a range of cardiovascular and thrombotic diseases in some human populations, underscoring the importance of its timely inactivation for maintaining health. The breadth of PAH's actions is truly remarkable, making it a critical lipid mediator to understand across numerous physiological and pathological contexts.
Okay, section 7. Therapeutic uses of eicosanoids and their modulators. So we've covered the intricate biochemistry, the broad physiological roles, and the complex pathological actions of eicosanoids and PAF in exceptional detail. Now let's bring it full circle to how we actually use this knowledge therapeutically, harnessing or blocking these powerful body signals with drugs to treat disease. What are the main ways we inhibit or antagonize these systems?
This is truly where the rubber meets the road in pharmacology, translating fundamental science into patient benefit. As we discussed at length, NSAIDs are widely used for their anti-inflammatory, analgesic pain-relieving, and antipyretic fever-reducing effects. Low-dose aspirin in particular is a cornerstone for cardiovascular protection, primarily by irreversibly inhibiting COX-1 in platelets to reduce clot formation. For leukotriene pathways, leukotriene antagonists are clinically useful in treating asthma, especially for mild to moderate severity. These include drugs like zafirlukist, montelukast, and prenlukast, which block the actions of cysteine leukotrienes at their receptors. They're also the mainstay of treatment for aspirin-exacerbated respiratory disease, or AERD, given the underlying dysregulation of leukotriene production in that adverse response.
So for asthma and AER, the leukotriene inhibitors are key therapeutic tools. What about drugs that target other specific eicosanoid receptors?
Right. FPR agonists, which act on the prostaglandin F2 receptors, are effectively used in the treatment of open-angle glaucoma, as they help decrease intraocular pressure by increasing fluid outflow from the eye. EPR agonists, which act on prostaglandin E2 receptors, are specifically used in obstetrics to induce labor and to ameliorate gastric irritation caused by NSAIDs, offering a protective effect for the stomach lining. DPR1 antagonists have also been explored for offsetting the facial flushing associated with niacin treatment, showcasing a very specific and targeted therapeutic application for a common side effect.
Beyond inhibition, we also use prostanoids and their analogues directly, administering them to patients despite their generally short half-lives and potential for systemic side effects. What are some of those remarkable clinical uses?
You're right, there are therapeutic challenges, as many endogenous prostanoids have short half-lives in circulation, and systemic administration can cause significant adverse effects like generalized pain, flushing, and diarrhea. Nonetheless, several prostanoids are of great and specific clinical utility. For labor induction and therapeutic abortion, PGE2, PGF2K, and their synthetic analogues are widely used to induce labor at term and to terminate pregnancy by promoting powerful uterine contractions. They also facilitate labor by promoting the ripening and dilation of the cervix, making delivery easier. Examples include dinoprostone, which is synthetic PGE2, or misoprostol, a synthetic PGE1 analog, both used for cervical ripening and labor induction, and as abortifacients in the second trimester. Misoprostol, when combined with the antiprogesterone mifepristone, RU486, is highly effective for medical pregnancy termination. For controlling postpartum hemorrhage, severe bleeding after childbirth, carboprostomethamine, a PGF2A analog, is used to induce second trimester abortions and powerfully contract the uterus to stop bleeding unresponsive to conventional methods.
That's a powerful and crucial set of tools for obstetric care. What about maintaining the patency of the ductus arteriosus in neonates with certain congenital heart defects?
For this critical pediatric application, PGE1, or alprostadil, is highly effective. The ductus arteriosus in neonates is exquisitely sensitive to vasodilation by PGE1. Maintaining a patent ductus may be hemodynamically important in some neonates with specific congenital heart diseases, allowing blood to bypass critical blockages and providing temporary patency until corrective surgery can be performed. A notable adverse effect, however, is apnea, or temporary cessation of breathing, which is observed in about 10% of treated neonates, particularly those weighing less than 2 kilograms at birth, often requiring ventilator assistance.
And earlier we discussed misoprostol for gastric cytoprotection for stomach ulcer prevention.
Yes. Misoprostol, that synthetic PGE1 analog, is approved for the prevention of NSAID-induced gastric ulcers, and is about as effective as the proton pump inhibitor omeprazole in this role. It helps bolster the stomach's protective barrier and reduce acid. However, it's absolutely crucial to remember that misoprostol is contraindicated in pregnant women or those who might become pregnant due to its potent abortifacient effects, highlighting the immense importance of careful patient selection and contraindication awareness.
What about for erectile dysfunction? Was it used there?
Yes, PGE1, alprostadil, given as an intracavernous injection or urethral suppository, used to be a prominent second-line treatment for erectile dysfunction due to its vasodilatory effects on penile arteries. However, it has largely been superseded by the oral phosphodiesterase 5 inhibitors, such as sildenafil, tadalafil, vardenafil, and enamel, which are now the preferred treatments due to their ease of administration and efficacy.
And for pulmonary arterial hypertension, or PAH, a severe and progressive lung disease?
For pulmonary arterial hypertension, PGI2, or epoprostenol, is used as long-term therapy via continuous intravenous infusion. This significantly improves symptoms and hemodynamics, potentially delaying or even precluding the need for lung or heart-lung transplantation in many patients. However, it is rapidly metabolized, requiring continuous infusion. Several orally available PGI2 analogues are also used clinically for PAH, offering more convenient administration. These include iloprost, which specifically targets lung vasculature and can be inhaled or given intravenously, and treprostinil, which has a longer half-life and can be delivered by continuous subcutaneous infusion, intravenous infusion, or orally. More recently, selexipag was approved. It's the first PGI2 analog with a non-prostanoid structure, an oral selective IPR agonist, and it has a longer active metabolite half-life, offering a novel oral option. Common dose-limiting adverse effects for these drugs include nausea, vomiting, headache, hypotension, and flushing.
Iloprost may also increase bleeding risk when combined with anticoagulants or platelet inhibitors, and selexipag is contraindicated with strong CYP2C8 inhibitors, so careful drug interaction management is essential.
Finally, let's revisit glaucoma, specifically how eicosanoids are used in its treatment.
Latanoprost, a stable, long-acting PGF2 derivative, was the pioneering prostanoid used for glaucoma. Similar prostanoids with ocular hypotensive effects that decrease intraocular pressure include bimatoprost, tafloprost, and travoprost. These drugs act as agonists at the FPR and are administered as ophthalmic drops, effectively decreasing intraocular pressure by increasing aqueous humor outflow. Common adverse effects may include changes in pigmentation, particularly increased iris pigmentation, which can be permanent, and increased eyelash growth, known as hypotrichosis. Upper respiratory tract infections can also occur in about 10% of patients. Latanoprost has also been creatively combined with other moieties, such as butanediol mononitrate, which is a source of nitric oxide, and a newer drug called latanoprostene bonode, and with Rho kinase inhibitors, showing ongoing efforts to enhance their efficacy and reduce side effects through combination therapies.
Given the excitement around PAF's discovery and its potent effects, what about PAF receptor antagonists? Have they found clinical utility, or has that been another challenge in this complex field?
Unfortunately, despite significant experimental efforts and the existence of several selective PAF receptor antagonists that effectively inhibit PAF actions in vivo and in vitro, none has proven clinically useful to date. This has been a source of disappointment in the field, highlighting the challenges of translating promising preclinical data into effective human therapies. Even inhibiting PAF acetyl or PAF-AH, the enzyme that breaks down PAF and also functions as a lipoprotein-associated PAA2, was studied as a maneuver that might elevate endogenous levels of PAF for potential therapeutic benefit. However, trials of an inhibitor called daraplebib failed to establish either clinical efficacy attributable to eicosanoid suppression or an adverse effect profile attributable to increased levels of PAF. This really underscores the immense challenges of targeting these complex lipid pathways in the way that translates into effective and safe treatments, reminding us that biological systems are often far more redundant and interconnected than we initially perceive.
Right. So wrapping this incredibly detailed deep dive up.
What's truly fascinating here, as we've explored in depth, is how these seemingly simple lipid molecules, once activated, become such sophisticated and precise communicators. They act as autacoids, local hormones, that impact virtually every physiological system, from the beat of your heart to the very structure of your bones and your body's nuanced fight against infection and cancer. It's a profound testament to the elegant and often surprising complexity of cellular communication, revealing how life is orchestrated at the molecular level.
So what does this all mean for us as we strive to master this information? We've taken a truly comprehensive expert-level deep dive into eicosanoids and PAF, from their historical discovery and incredibly intricate biosynthesis, through their detailed mechanisms of action, their widespread physiological effects across nearly every body system, and finally, their complex therapeutic applications. We've seen how a nuanced understanding of these pathways can lead to life-saving drugs, but also reveal unexpected challenges and side effects, like the cardiovascular risks associated with some anti-inflammatory medications. It truly illustrates that in biology, there are always deeper layers of interaction and balance to uncover.
Indeed. And this deep dive really emphasizes that mastering a topic means appreciating not just the what these molecules do, but the profound how these molecules are precisely made, how they signal with such specificity, and why even subtle changes or imbalances can have such profound system-wide effects throughout the body. Understanding this fundamental lipid language is key to unlocking so much in medicine, biology, and the future of therapeutics.
Think about it. Given their central roles in inflammation, immunity, and pain, how might our growing expert-level understanding of these intricate lipid networks continue to reshape and revolutionize treatments for chronic diseases like cancer, autoimmune disorders, or even future viral pandemics? What stands out to you about these incredibly versatile 20-carbon messengers now that you've explored their world in such depth?